So I'm now pleased to call upon Dr. Andrea Kirkwood for her presentation. She'll be having a slide presentation on screen. After her presentation, we do have 5 minutes or so for questions, which we welcome from directors and members alike. Thank you. Dr. Andrea Kirkwood.
Rookie move. So hopefully everyone can hear me. Thank you for attending the AGM this evening. Yes, it probably would have been a lot nicer to be meeting in person, but hopefully this will be kind of a nice consolation prize. So I recognize now that depending on how you have your Zoom set up, you may see people. If there is a way to get rid of, if you don't want to see me, I think you can make that disappear. But I don't have the control, so I'm going to be saying next to Tim. So before I say next, I just want to point out that, and thank you to the Scugog Lake Stewards. I got this picture from one of your Facebook postings, that as you can see with all the logos on the bottom of the screen there is a lot of organizations and people involved in the study that I'm going to be talking about tonight. OTF standing for the Ontario Trillium Fund study, and really it gave us an opportunity to do a very extensive assessment of the lake in terms of both investigating walleye habitat conditions and just overall lake health. So next. Okay, so before I proceed with my presentation, I would like to give a land acknowledgement. We would like to acknowledge the lands and people of the Mississaugas of Scugog Island First Nation. We are thankful to be welcomed onto these lands in friendship. The lands where we are situated on are covered under the Williams Treaties, and they are the traditional territory of the Mississauga, a branch of the Greater Anishinaabeg Nation, including Algonquin, Ojibwe, Ottawa, and Potawatomi. And these lands remain home to a number of Indigenous nations and people. And that logo is actually was designed by an Indigenous member of the Scugog Island First Nation community. Okay, next. Okay, so I only have 15 minutes, and I think I might be really pushing it because we have a lot to go over here. But I really just pulled. Everyone contributed, and you'll see. I'm going to show you all the partners involved. But so there's in no way could I have presented everything from a 3-year intensive study. But just to let you know that the overall objective of this 3-year study was to understand some of the factors that may be contributing to the decline in walleye, while also improving our understanding of overall lake health in Lake Scugog. And so thank you to the Ontario Trillium Foundation for supporting our Ontario Trillium Grow Grant, which if we go back in time to, I think it was around December 2016, Josh Thiempont and Ron Porter and even Barbara, I think, was involved at that point, were really instrumental in pulling that proposal together. Next. So before I proceed, I really want to highlight the structure of this project because it wasn't just done by a handful of people. There was many people, many chefs in the kitchen, shall we say, but we all had our job, and I think it really worked well. And in many instances, we were kind of meeting each other for the first time, when coming together to get this project going. So as you can see, the Ontario Trillium Fund supports community groups. And so the Scugog Lake Stewards should take full credit for really being the overarching project managers of this study and really helping out with the logistics. So Dr. Ron Porter, which I was hoping he might be attending tonight, Barbara Cartine, Colleen Dempster, Chuck Haru, Tyler Harrington, and Chris Hockley. These are all folks, and I'm sure I've probably missed a few people, but from just memory over the last 3 years, just being really instrumental in logistics and helping us with study design and so forth. So that's why they've been included as research partners. Now at Ontario Tech, where I'm the professor, the key kind of project leads were my graduate students, Tyler Harrell-Lyle and Aaron Smith, who are attending tonight. At York University, Dr. Jenny Carosi and Dr. Josh Thienpont, who also live in Port Perry. At Kawartha Conservation, Debbie Belika, who just recently left that role and is now working at Conservation Ontario. So congratulations, Debbie. But we'll miss you at Kawartha Conservation. And Brett Trigano, who I think is trying to get into the Zoom meeting here. I sent him the link. And then finally, the Ontario Ministry of Natural Resources and Forestry were involved at the very beginning. Lisa Solomon was our contact there, and she left the role, I think, within the first year of the study. And, you know, without going into the full details, I think there's a lot of restructuring and so forth happening. They weren't as involved in the project, so I don't have any results to show from them per se, but we're still, you know, hoping to engage with them in the future. So I'm just going to be presenting mainly the projects that were executed by Ontario Tech, York University, and Kawartha Conservation. Next. So the first project I'm going to talk about was led by Tyler Harrell-Lyle, who is a PhD candidate now. And he actually started off as an undergraduate student. And so we're actually sneaking in some 2016 data into this presentation. So 4 years of data on these sites that you can see in the right-hand side is the watershed map. So that what you see is starting off with the watershed, and then you see the different colors, and you see basically comparing agricultural, natural, and developed land. So the developed land is in the bright red. So think of urban development. And then you see the sites distributed around the lake. And so these 12 sites, although they look like they're right on the shoreline, we consider them more offshore. They're based on historic maps of walleye spawning areas. And so these represent monthly samples. Next. So what I want you to see, so these look very sciencey and busy, but really just step back and focus on where I've delineated West Arm and East Arm. And the numbers on the bottom represent the sites that you just saw on that previous map. And so all the little dots really represent the data are noisy. And so to clean up the data, we create these box plots. And really, just think of the box plot, how tall it is represents how much range there is, going from low values to high values. And so I just really want you to look at the big bars and how high they get. So in chloride, those that are getting up to around 400 milligrams per liter, that's high, versus the East Arm, where you see that they're generally lower, okay? So West Arm, high chloride, and then total phosphorus is another parameter that we measure that also tends to be a little bit higher in the West Arm. Okay, so if you recall the West and East Arm of Lake Scugog, West Arm, the south end, Port Perry exists, and then the East Arm is a little bit deeper and less developed. Now, why am I focusing on chloride and total phosphorus? Well, chloride, maybe I would have run a poll if you were in my class, but road salt. And when we think of road salt, what do we use road salt for? Well, we usually like to de-ice our roads and our sidewalks so that people don't get into accidents and fall on the ground and hurt themselves. So there's lots of road salt application happening wherever there are roads, high densities of roads, as well as driveways and parking lots. And not surprisingly, we find chloride is really high so site 1 and 2 is what we would consider the Port Perry area, okay? Now with phosphorus, we usually think of phosphorus, which is a really important nutrient for algae and aquatic plants. And if you remember reading the Lake Scugog Lake Management Plan from 2010, it talked a lot about phosphorus because phosphorus is really the key limiting nutrient. If we can keep phosphorus lower, we'll have less weeds and we'll have less algae. And so what's happened in the last 4 years, which these data are basically showing data across this time frame, is that the West Arm, which tends to be a bit more developed, actually has more phosphorus than the Eastern Arm. And I find this really fascinating because we usually think of agriculture like cow poop and fertilizers as being an important source of nutrients like nitrogen and phosphorus. But what we found in this study is that the urban areas are actually significant contributors of nutrients as well as chloride. And so when you have high chloride and high phosphorus, this contributes to poor water quality. Okay, next. So you'll look at this and you're like, whoa, what am I looking at? By no means do I expect you to read the little tiny print. What I love about these figures that Tyler made, and I asked him, ooh, what's with the pukey green color, brown, and Tyler was one step ahead of me. He's like, well, this is accessible colors for people who are colorblind. So kudos to you, Tyler. But all I want you to do is maybe you can step back a little bit. Okay, so you see these panels. You see a 2016 panel, a 2017 panel, 2018 panel, 2019 panel. Okay, so those represent the 4 study years. Now you'll see these vertical bars of different colors. And so each color represents a particular species of aquatic plant. So Myriophyllum spicatum is one of them. So then you have Utricularia, which is that carnivorous plant in the lake. It's all good. But I just want to give you an idea. What do the colors mean? The more variety of colors you see means there's more plants, different types of plants in the lake. And so really, as you go across, so each bar represents a site and a date. But really, I don't want you to get lost in the weeds, pun intended. I want you to step back and really look at 2016 to 2019. What do you notice is happening in the lake? You go from lots of diversity in 2016, relatively speaking, and then 2017, whoa, things change. There's that pukey green color that's becoming really, really abundant and dominant. Again in 2018, then again in 2019. It seems to have really taken over. And if you want to guess what that species is, it's actually it's not actually a plant. It's what we call a macroalga, known as a charophyte, and its scientific name is Nitolopsis obtusa, also known as starry stonewort. And so starry stonewort is gaining a lot of attention in the last couple of years because it's starting to take over throughout the lakes connected to the Trent Severn Waterway, including Lake Simcoe and Stony Lake, because I think Pat would be interested in that. So what I'd like to say, kudos to the Scugog Lake Stewards for being we are the first group, a research group to study starry stonewort, say that fast 3 times, in Ontario. And so that's actually what allowed Tyler to expand into his PhD and study this invasive species. So for those of you who aren't familiar with starry stonewort, it is originally from Eurasia, so you can find it. Its native areas are in Great Britain, in parts of Europe, and Japan. And so it was introduced about 40 years ago into the Great Lakes, and now it's spreading into the inland lakes. Okay, next. Trying to be mindful of time. I could go on and on. Okay, so microcystis, what is this species? Well, when you think of those blue-green algae blooms that started to happen in the lake around 2016, so serendipitously, Tyler and his crew were out in the lake. The stewards gave some seed money to start an initial study that helped us get the larger grant. And you can see, if you look at the lake and just think of, this is what we call a heat map. So where you see a dense, bright red dot, that means that that's probably indicative of a microcystis bloom, so a blue-green algae bloom. So in 2016, on average, those are the sites in the lake where there was high levels of this microcystis. And microcystis is a type of blue-green algae, also known as cyanobacteria. We want to be properly scientific about it. And it has the potential to produce toxins. That's why we care about it. And it doesn't look nice. It looks like pea soup when it's blooming. Now, what I want you to see from here is that it's not throughout the entire lake. It's only in certain embayments. So microcystis is kind of a sensitive creature. It needs certain conditions. And as you go to 2017, 2018, 2019, every year there were instances of bloom events, but very localized, okay? Now, you can't really see it here. It's showing us that microcystis gets around, but it tends to be more of a problem, again, down in the Port Perry area. And I'll explain a little bit more why that seems to be the case. Okay, next. Okay, so that did, a very rudimentary job of Tyler's 4-year study in Lake Scugog, but we'll move on to Erin Smith. So she's another PhD candidate in my lab, and she's with us tonight. And she started as well as an undergraduate student, as a summer student on the Lake Scugog project, and it has now expanded into a much larger project across the Kawartha Lakes. So again, the stewards are instrumental in graduate training for master's students, so undergrad, master's, and now PhD. So her project is in partnership with Debbie Belika. So Debbie Belika was the brainchild of this study, nearshore study, because she really recognized, with working with community groups and working with shoreline property owners, that the nearshore was pretty much being ignored when it comes to monitoring programs. Usually monitoring programs, the Lake Partner program you're probably familiar with, you go out into the lake, usually the middle of the lake, to take water samples. And yet the nearshore is where all the action's happening. That's where you have, aquatic plants, because the water tends to be shallow, so aquatic plants can establish. Great, aquatic plants are great habitat for fish. They tend to be nurseries for fish. And yet, when you think of stormwater runoff, remember that road salt I was talking about? Where do you think it goes? It runs right off of the land and right into the lake. And so this is a key area we wanted to get a better handle on its current conditions. And so what you see here on the right-hand side is another one of these maps where Erin was able to overlay her data of chloride. And again, where do you see this hot spot for chloride, really high chloride concentrations in the Port Perry Bay area? And so those sites down there are showing that chloride is a significant issue, and chloride is toxic to fish and other aquatic life at certain levels. And we certainly found that sometimes, depending on the date, it would get pretty close to toxic levels, what we call the chronic 30-day levels in the Port Perry Bay area. Next. So here's the same map, but now we're looking at phosphorus, okay? So phosphorus, it gets around a little bit more in the lake. It's a little bit more distributed, shall we say. But when you think about it, phosphorus comes from many sources. Phosphorus can come from when people fertilize their lawns. Phosphorus can come from leaky septic systems. Phosphorus can come from fertilizer application, and phosphorus can come from cow poop and pig poop. So lots of sources of phosphorus, and this is probably why we see phosphorus everywhere. But what we're mostly interested in is, where is phosphorus high? Because phosphorus isn't like a toxic compound. It is required by all organisms, even our cells. We need it in our bones and our DNA. But when there's too much of a good thing, algae go gangbusters. And this is why we're finding that phosphorus is an important driver of microcystis blooms in the lake as well. So again, Port Perry Bay area, very high levels of phosphorus. And then interestingly enough, over on the east arm, there's a potential important source of phosphorus over there as well that we're going to be looking into a little bit more. But right now, Erin and I and Debbie are working on a paper that we're pulling together where Erin was able to look at all the different levels, spatial levels of influence in the lake. So just to try and simplify it, remember I was telling you all these different sources? So say if there's a farm way up in the north end of the watershed and it's got, it uses lots of fertilizer. Well, that fertilizer could eventually end up in the lake because it'll get the heavy rainstorm, the water rushes into a tributary is a creek, and then that creek flows into, say, the Nonquan, and then the Nonquan River flows into the lake. So it can eventually get there. And so with her analysis, what we call geographic information systems analysis, she was able to show that when it comes to chloride and phosphorus, it's not that farm up on the north end of the watershed or in, far away from the lake, catchment scale, but it's actually at the very close local scale, which means that people living near the lake are having this impact on the lake with respect to chloride and phosphorus. Next. So I'm going to shift gears to the York University crew. So Josh and Jenny, I don't know if Josh is here, but Jenny's in attendance tonight. And one of their projects was to look at oxygen levels during winter. And this is actually a big field of study now across North America, this what we call winter limnology. Limnology is the study of inland waters. And we know that, fish are overwintering in the lake. And if oxygen gets too low, because when you have too much plant material and it decomposes, the bacteria can actually suck up the oxygen, so consume it, because they're just like us, they need oxygen to breathe. And when oxygen gets consumed, you can get what we call anoxic or hypoxic events that can kill fish. And many shallow lakes, especially in the prairies, fish die in the wintertime because of this process. So we, or I should say Jenny and Josh wanted to investigate this. And next. And there is a picture, I think that's Jenny in the background there, but there's Josh in the foreground holding the, looks like it's the probe for sure. I guess he's calibrating it. And he really just wanted to point out here that they sampled on the coldest day of the year, but I'm assuming the red circle is showing that that year it wasn't very cold. So it wasn't very safe to go out on the lake. And so they really only had one sampling event, a date that they went out that was safe to go out. But the good news is that when they did measure the oxygen, it was actually pretty high. So yay, because that means the fish aren't being stressed out with no oxygen. And what produces oxygen under the ice? Photosynthesis. And it means the aquatic plants were getting light from, so there mustn't have been a lot of snowpack cover to block out the light. And so photosynthesis was likely supporting that. So that's actually a really good, something good we wanted to find out. Next. So the next topic, which I think is really super interesting, is this an approach to limnology, which is known as paleolimnology. So as the name suggests, paleo meaning ancient. Think of paleontology, people who study dinosaurs in ancient life. Paleolimnology is a way to take cores. So I don't know if you can see, if hopefully you don't have people's faces blocking it, but on the right-hand side there, you can see Josh holding a large core, and it's see-through. So you can actually see a sediment profile. So that gets deployed at the bottom of the lake and goes deep into the sediment. And when it pulls it out, the top of the core is the most recent sediment. And as you go down deeper and deeper, you're going back in time. And so you can actually see with the color change there represents when Lake Scugog was initially flooded back in the 1800s. Isn't that really cool? And so this type of technique means that we can look at pigments, we can look at animal parts like zooplankton and chironomids, and we can reconstruct past environmental conditions. Okay, so Jenny and Josh did coring in Port Perry Bay. I think it was also connected with coring that had been done there as part of the LSEP project, and then a deep basin in the Eastern Arm. Next. So there's that core. So the left-hand side, you see this cartoon diagram, and you see, an illustration of that core I just was talking about that Josh had shown. And what it's doing is you can see that it zooms out the possibilities of looking at all those different types of compounds and organisms that we can make sense of, because we know certain organisms are only found under certain conditions. And so we basically let the organisms tell us what the conditions were like back then. So algal pigments are just one of those compounds that we can measure. And so next. Okay, so I just want to draw your attention to a particular pigment known as myxoxanthophyll, which is associated with cyanobacteria, and I believe microcystis in particular has this pigment. And what's really interesting is if you look below the red line in that column of myxoxanthophyll, you'll see that each black bar represents the presence of microcystis in the past, going all the way back into the early 1800s. And yet in the 1990s, it disappears. So this is interesting. because I just showed you a few slides back that we're having microcystis blooms now in the lake. Next. So we basically, it's like being a bit of a detective, and I think in a way paleolimnology is like being a forensic scientist. So Jenny's lab has been able to show that at least we have an idea, because a big question was, well, is this occurrence of microcystis blooms, is it new? I asked the stewards, I talked to Kawartha Conservation. There really wasn't, I kind of looked in the literature, no reports of microcystis blooms in the last 20 years. But now we have evidence to show that there hasn't been any blooms in the last, since last 30 years. So we suspect that the recent reemergence of microcystis is probably related to climate change and urbanization. Next. So this is the final project I'm going to talk about. I only included those yellow circles to just highlight the sites, because there's a lot of them and I found it hard to see them. So I just thought I might help you guys out just by really delineating where the sites are, these tributary sites. So if you recall, tributaries are creeks or streams, rivers, anything flowing that they contribute to the lake. So they flow into the lake. And we know that walleye, they can spawn in the lake, but they can also spawn upstream in tributaries. And so we were able to use some money that was in the OTF grant to go towards this study in 2019 that Brett Trograno had led. And he was able to, and his team were able to look at 111 stations within 11 tributaries. And they looked at various factors that are important for profiling habitat quality for walleye spawning. And next. And the key findings that Brett found was that, here's some more good news. There are many tributaries in the watershed that could be suitable walleye spawning habitat. However, the key habitat factor was substrate. So oxygen levels, depth, all of these, the other factors that they looked at were all at kind of ambient conditions, they're fine. But where substrate wasn't ideal, it was basically is kind of make or break whether a site was good for spawning. And so basically he concluded that the most degraded sites, so poor substrate, so silty, mucky, not nice kind of cobble, usually nice stone cobble substrate is ideal. But in the silty sites, they tended to be the urban streams like Coppers Creek in Port Perry and Sucker Creek in Lindsay. Okay, so these findings will inform future walleye population monitoring. So I would say this kind of 1 year study is doing so much for informing future walleye population studies as well as rehabilitation projects as well. Okay, next. Okay, so I know that was a lot of information, but I'm hoping that it's giving you kind of an overall picture of Lake Scugog, that there's some concerns, but there's also some good news as well. Okay, so next. We are able to conclude that in a watershed that is largely agricultural, urban development, which is, on an area per area comparison, much smaller, but orders of magnitude more significant in its impact. And we've been able to delineate that through 3 different independent studies. So with Tyler's study, with Aaron's study, and with Brett's study, urban areas are associated with degraded habitat characteristics. Next. Aaron was able to show in her nearshore study that local land use activities. So think about how shorelines are being managed. So are they naturalized? Is there potential septic leaking issues? Overuse of fertilizers? This is actually, so in one sense, we can think of, okay, well, that's a problem, but it's actually an opportunity for management. And rather than waste resources targeting farmers upstream, let's target where a lot of this is happening at the local level. Next. The winter oxygen levels, yes, it was one one year and basically one event that was studied, but I would say let's take it. That's pretty encouraging. Next. And these microcystis blooms, of course, as scientists, Jenny and I are very interested in pursuing this a little bit further, but it's with the research that we've done with this project and then research that's also happened in other projects in my lab, we're starting to make a connection between chloride and microcystis and phosphorus. That's the trifecta. High phosphorus, high chloride, and microcystis blooms tend to occur. Okay, so that's again connected with urbanization and climate change, which unfortunately we can't, there's not a lot we can do with controlling climate change, but we can certainly adapt to it. Next. And then finally, another good news story is the tributary sites, that there's lots of potential for whether it's restoring population numbers, working with OMNRF on the walleye population in the lake, and noting that if there's going to be efforts to restore sites, they should be probably targeted at the urban tributaries. Okay. Sorry, next. So without further ado, I just wanted to list, and I apologize if I've missed any names, but I'm just, again, these are people that helped out along the way. Some have come and gone, but certainly they aren't forgotten. And there's a whole crew of summer students. Kawartha Conservation also had volunteers. We wouldn't be able to have had the nearshore study without community volunteers. John Mackey at Port Perry Marina deserves a big shout out. He always came through for us with many different challenges. And then finally, Jen Sutterton, who made these wonderful videos as part of our OTF deliverables. And I encourage you, I can't believe that some of these videos that are so amazingly produced only have 75 views or something. So we really need to start marketing these videos a little bit more. And then finally, thank you to the Ontario Trillium Foundation. And that's it.
Excellent.